This is the fall final exam review. I'm going over it with you so that you can study more effectively. Part 1 is identifying the significance of these diagrams to biological systems. So this right here is the element carbon. And carbon is important because it's an essential, substantial portion of all living things. It makes up about 19% of the human body. And it's the basic unit that organic molecules are built around. So the element carbon can make 4 bonds, and they are able to make those 4 bonds to create proteins, carbohydrates, nucleic acids, and lipids. Next diagram is water. And water is important because it's an almost universal solvent. Reaction in photosynthesis is a reactant in photosynthesis, and it is a product in cellular respiration. And then, of course, one essential property of water is the next diagram here, hydrogen bonding. So hydrogen bonds, remember, are weak attractions between the partial positive charge of a hydrogen molecule in water and the partial negative charge of oxygen in a water molecule that is close to it. So these hydrogen bonds are going to provide the unique properties of water that we see, like cohesion, where water molecules stick to each other, and adhesion, where they stick to other surfaces through those hydrogen bonds. And it's going to cause things like the recently learned phenomenon, transpiration in plants, to happen, where we're drawing water up through the roots, through the stems, and out the leaves. The opening is called stomata. Our next diagram, lipids. This is actually a triglyceride. Sorry, that should be TRI. I don't know why it says that. And remember that lipids are long-term energy storage in organisms. So if they cannot use the carbohydrates at that point in time, they'll turn it into a lipid so that it can be stored later. The next diagram here is glucose. And glucose is important. It's an essential carbohydrate, and it's used as direct or fast energy in cells because, remember, this is what is used in cellular respiration. That's glucose. Next is the amino acid. So the amino acid has a central carbon with an amine group on one side and a carboxyl group on the other side. And then it has this R group here, and the R group is variable, meaning that it changes. And so there's 20 different amino acids in use in living organisms. The amino acids come together to make proteins, and these different side chains are going to contribute to how proteins fold. And remember, whatever different shape a protein has is going to be what it does. So the fact that we have all these different side chains in the amino acids allow for the different jobs that proteins are able to perform. This diagram here is denaturation. So denaturation, remember, is when the shape of the molecule changes, and this happens because the hydrogen bonds and other molecular bonds are changed due to a high temperature and a dramatic change around them in pH, which causes the molecule ultimately to lose shape. And if proteins and enzymes lose shape, they're no longer able to do their job because their shape is very specific to what they do, how they work, where they work. The next diagram is the enzyme substrate complex. So A is the enzyme, B is the substrate, and then the active site is where they attach. So remember that substrates are going to bind to enzymes at the active site. The substrate is a very specific shape. The active site is a specific shape, and the enzyme carries a very specific shape. So once the substrate attaches, the reaction happens, and we either put molecules together or break them apart. We get a product out of it, but the enzyme does not change. The enzyme stays the same, and enzymes are recycled over and over again. Here, this is a phospholipid, is the basic component of cell membranes. The phospholipid has a phosphate head, which is hydrophilic. It is attracted to the water. Then it has 2 fatty acid tails, which are hydrophobic and are repelled by the water. So when you see a phospholipid, in the cell, it's going to be in a bilayer, meaning 2 layers. Remember, like our little jellyfish hands that we made. The heads are going to face the outside, and then inside the cell membrane, the fatty acids face each other in that hydrophobic environment. And this is what causes barriers between the cell and the outside world. The next diagram is a diagram to represent diffusion, which is the movement of particles from high to low concentration. This is going to be necessary for movement of water, obviously in the case of osmosis, but also if you remember, one of the scenarios we learned during cell transport was red blood cells in the lungs. So oxygen is able to travel into red blood cells from the air we breathe into the lungs, while at the same time carbon dioxide is leaving our red blood cells and being leaked out into the lungs so that we can breathe carbon dioxide out. So that's all a function of diffusion. This is a picture that represents crenation, which is what happens when a cell loses mass or it loses water because it has been placed into a hypertonic environment, meaning there's a lot of solute outside of the cell. Okay. This diagram represents tumor, cancer. Cancer is uncontrolled cell division. So eventually we get to the point where we have a mass of cells that is growing like crazy, dividing like crazy. There's no regulation. This is usually due to mutations in oncogenes and tumor suppressor genes. These are the types of proteins and genes that regulate cell division. Our next diagram is a nucleotide, which is the building block of DNA and RNA. It's the building block of our nucleic acids. Depending on the type of nucleic acid, the sugar and bases might change a little bit. So the sugar in DNA is deoxyribose sugar, but in RNA it is just ribose sugar. And then the nitrogen-containing base in DNA will be either A, T, G, or C, adenine, thymine, cytosine, or guanine. If it's RNA, it will be A, U, C, G, adenine, uracil, guanine, cytosine. And then remember that the order of the bases that we see in the middle of the double helix is going to govern the protein that is made, which is going to give us our individual traits. Okay. All right, moving on, anaphase. In this phase, this happens before telophase, after metaphase. Sister chromatids are separated in the cell, and this occurs so that we can make sure that equivalent amounts of DNA are split between the daughter cells. And this is important because each daughter cell needs to have their own copy of DNA. Metaphase is another phase that happens during cell division. It happens right before anaphase, and the chromosomes are actually positioned at the equator, and checkpoints actually ensure that the chromosomes are in the right place so they can be split evenly. And this happens after prophase. Remember PMAT, prophase, metaphase, anaphase, telophase. Pass me a taco to remember the stages of M phase. Next we have virus. Remember that a virus is a non-living particle that disrupts homeostasis of cells. So that's why we learned about them in biology, because they have an effect, large effect on living organisms. All cells are able to become infected with viruses, whether they're prokaryotes or eukaryotes. Remember that viruses are only genetic material and protein. So the genetic material can be DNA or RNA. And the protein component of viruses is called the capsid, and it surrounds the genetic component inside. Some viruses actually have a lipid envelope that they took from their host cell when they budded out of the cell. So that's only some viruses, not all viruses, and those come from their host. Remember, viruses are not living. They don't respond to the environment. They don't grow. They don't develop. They don't need energy. They don't do any of the things that normal living cells do. So they are not living. Next, we're going into chloroplasts. Remember that chloroplasts are double membrane bound organelles. So if they're an organelle, they're only found in eukaryotic cells. What they do is they house the chlorophyll for photosynthetic organisms so that they can absorb sunlight more efficiently. So chlorophyll is a pigment that helps convert radiant energy into the bonds of glucose, into chemical energy. And that's the chloroplast. Then we have the mitochondria, which is also an organelle that is double membrane bound, and it's in eukaryotes. And it's going to carry out cellular respiration, which we're going to convert that sugar that was produced into ATP. All life is going to do cellular respiration, but not all living things have mitochondria. Just like there are some bacteria that are able to carry out photosynthesis, but they don't have chloroplasts. What they do contain, though, is chlorophyll that allows them to carry out photosynthesis. Organisms that are able to carry out cellular respiration without a mitochondria have those enzymes and protein complexes embedded in their one membrane that surrounds the cell and allows for that to happen. This diagram represents ATP and ADP, the fact that they cycle in the cell. So remember, ATP stands for adenosine triphosphate, three phosphate molecules put together. What the cell will do is it will take the phosphate molecule off of the ATP, and when it does that, it releases energy and creates an ADP molecule. And then the cell will take the ADPs, recycle them, and reattach a phosphate on so that we can reuse those molecules. So the cell will cycle back and forth. ATP, ADP, ATP, ADP, making energy, using energy, making energy, using energy. Okay? Okay, part 2 vocabulary. So this right here, no energy, water movement of solvent, osmosis. This one requires energy, low to high concentration against the concentration gradient, sodium potassium pump, active transport. This one, no energy, equalized diffusion. This one, facilitated diffusion, high to low. The difference between facilitated diffusion, though, is that it uses a protein to carry out the transport. But don't forget that facilitated diffusion uses a protein carrier. Endocytosis is going to take large molecules into the cell using a vesicle. Remember, a vesicle is a little membrane-bound sac that the cell can produce when it needs to. So if we're bringing molecules into the cell, it's endocytosis. If we're taking them out, it is exocytosis. The next group of words belongs to the cell cycle. So the nucleolus disappears. It's the beginning of mitosis. That is prophase. When we're pulling the chromosomes apart, we're separating the chromosomes to the poles. It's during mitosis, that's anaphase. When DNA is synthesized before mitosis, that is S phase. Remember, S phase is the second part of interphase. Interphase is G1, S, and G2. So growth checkpoints after DNA replicates before mitosis is G2. That's the last part of interphase before the cell starts mitosis. Then we have sister chromatids are together in the middle. We have these lined up here. That's metaphase. And then lastly, mitosis, two visible cells start to form. It's the last phase, that is telophase cytokinesis. Lipids are going to store energy in the cell, stored energy. They make up the cell membrane. They provide insulation. They allow for nerve impulses to happen, so the electrical signaling that your nerves are able to do. And they are also regulation hormones like steroids, and other fats are going to be lipid group of biomolecules. Then secondly, we have photosynthesis and cellular respiration, mitochondria, energy, structural support of RNA and DNA. Remember, there's a sugar and the sugar phosphate backbone of RNA and DNA. Those are going to be carbohydrates. Then next, we have the genetic code that is housed in the nucleus of eukaryotic cells. It's inherited. Those are nucleic acids, specifically DNA. Proteins are going to offer structure, support. They're going to make sure that chemical reactions happen. Proteins are expressed as a trait. They're going to provide defense, transport, and ribosomes are made of proteins. So remember, proteins do everything. All the work in the cell is done by proteins. Okay, this next group of questions, we're going to look at the statements and we're going to determine which one matches with which one. So remember that viruses are non-living particles. They do not have any homeostasis. They don't grow, they don't develop, they don't require energy. Then you have cells, which are the basic unit of life. All cells have a cell membrane. All cells have DNA. All cells have ribosomes. All cells have cytoplasm. So all cells are considered living. They respond to their environment. They grow, they develop, they reproduce, etc. And then both is going to apply to both living cells and non-living viruses. So number one, basic unit of life, that would be B, cells. 2, a non-living particle made of organic material, that would be A, a virus. And they're still made of organic material because they take stuff from their host cells, which are living, and their host cells are made of organic material. Then you have 3, made of organic material, that would be both are made of organic material. Number 4, genetic code could be DNA or RNA. Only viruses are capable of using RNA as their genetic code. Number 5 has an outer membrane and cytoplasm along with genetic material. That would be B, cells. Here's the cytoplasm. That's a key part of cells. Number 6 undergoes growth and development. Only cells can do that. Only life can do that. Number 7 does not need, obtain, or use energy, would be viruses. Number 8, reproducing independently is something that only living things can do. And number 9, if they require a host to reproduce, it's going to be viruses. Number 10, it's able to respond to the environment, would be only B cells. Same kind of thing here, but we're going to do prokaryote, eukaryote, or both. So remember, prokaryotic cells are simple. They have no internal organization. They're only unicellular. There's no internal organelles. Okay? And then eukaryotic cells are more complex. They have internal membranes and internal organization. They can be unicellular or multicellular. Or we're going to have both. That would be anything living would be considered both. So surrounded by a cell membrane, all cells, B. Perform cellular respiration. All living things have some sort of cellular respiration. Perform DNA replication. All living organisms have to replicate their DNA in order to make the next generation of cells. Whether that be growth and repair in multicellular organisms or reproduction in unicellular organisms. Number 4, use lipids, proteins, carbohydrates, and nucleic acids. That would be B. All living things use those biomolecules. Number 5, contain internal membranes, only eukaryotes. Number 6, performing photosynthesis is characteristic of both prokaryotes and eukaryotes, so that would be B. Number 7 are only unicellular, prokaryotes. Number 8 can be unicellular or multicellular, that would be eukaryotes. Number 9 are not specialized, prokaryotes are not specialized, so P. Number 10, make up tissues, E, eukaryotes. In order to make up tissues, you have to be able to form tissues. Only multicellular organisms have tissues, and only eukaryotic cells can come together to form those tissues. And then you have 11, the most abundant type of cell would be P, prokaryotic cells. Number 12 can be infected by viruses, that's both. Any living cell can be infected by a virus. Number 13, rely on DNA to transmit genetic information, that would be both. All right, so C works the same way, except, let's see, autotrophs, A, they make their own food. Heterotrophs, they have to seek out food, or both autotrophs and heterotrophs do this. So, number 1, perform photosynthesis, autotrophs only. Number 2, rely on carbohydrates for energy. All organisms rely on carbohydrates for energy. So B is essentially all organisms. 3, convert carbohydrates into ATP. All organisms do that. That's cellular respiration. Number 4, use ATP as fuel for cellular processes. All organisms do that. Number 5, convert energy. All organisms are able to do that. But autotrophs are able to take sunlight energy and turn it into something we can use. Number 6, convert radiant energy into chemical energy, A. Number 7, contain pigment molecules. Remember, pigment molecules are for absorbing sunlight. Okay, so that would be only autotrophs, A. Number 8, perform glycolysis. That's the first step to cellular respiration, so that would be both. Number 10, perform light-dependent reactions. That would be autotrophs only, because that's the first stage of photosynthesis. Number 11, use electron carriers for fueling cellular processes. That's both. NADPH, NADH, and FADH2 are all molecules that are used as electron carriers, and they're a part of cellular respiration and photosynthesis. All right, the last section of this is identifying lipids, carbohydrates, nucleic acids, proteins, or organic molecules. So statement 1, made of hydrogen, oxygen, and carbon would be O, organic molecule. So for number 2, L and P are going to make up some component of the cell membrane. Number 3 would be N, monomers, the nucleotide. Number 4, P, protein catalyzed reactions, specifically enzymes. Number 5 would be P for protein, carries out all essential functions in a cell. Number 6, carbohydrates, specifically glucose. This is the direct source of energy for cells. Number 7, nucleic acids, N, are going to transmit genetic information. Number 8, organic molecules are essential for prokaryotes and eukaryotes. That's why they're organic molecules. Number 9, organic molecules are produced via dehydration synthesis. So remember, dehydration synthesis is where we're pulling the water out to make, so that would be all organic molecules, are considered polymers. That's all organic molecules. 11 would be lipids used for energy storage. 12, formed from amino acids would be protein. Number 13, broken down via hydrolysis. Hydrolysis means to break apart, and all organic molecules are broken apart that way. And then number 14, sugar phosphate backbone would be the nucleic acids. Speaking of nucleic acids, the diagram's here. Nitrogen base. Okay, remember that DNA uses A, T, G, and C, and then RNA is going to use A, U, G, and C. So there's your base. They both have a sugar molecule. DNA is deoxyribose, RNA is ribose. And then this right here, the P in the middle with the 4 oxygens around it, that's phosphate. So again, a different model, but the same thing. The nitrogen bases are in the middle, C, T, A, G. Phosphates are usually represented with circles. Sugars are going to be represented with a little house, a little pentagon. When you put them together, they make the backbone. So this is still the sugar, phosphate, sugar, phosphate, sugar, phosphate. It's just simplified to show just the backbone. And this the same way in this diagram here, sugar, phosphate, backbone. And the nitrogen bases are in the middle. And then don't forget about the hydrogen bonds that connect the base pair across the middle of the helix. Our next diagram, prokaryotic versus eukaryotic cell, you'll notice what they have in common. They both have a plasma membrane, they both have cytoplasm, they both contain DNA, they both have ribosomes. So, major difference, eukaryotic cells have a nucleus that has a membrane wrapped around it to contain the DNA, and a prokaryotic cell just has a region of its cell where the DNA hangs out, and that's called the nucleoid region, but it's not separate from the rest of the cell. Cell membrane. So here we have the components of the cell membrane. So the major component of the cell membrane would be the phospholipid. So here's the hydrophilic phosphate. It enjoys or is attracted to water, so that's why it's on the either side of the membrane facing the aqueous environment. And then in the middle here are the fatty acid tails, which are hydrophobic, and that's why they face each other on the inside of the membrane. Then you have proteins. If the protein goes all the way across, it's an integral protein. Channel proteins allow for things to pass through the membrane, and if it's only on one side of the membrane, it's a peripheral protein. Cholesterols help stabilize the membrane, making it more flexible or less flexible depending on what the temperature and what the needs of the cell are. Glycolipids are a combination of a glyco, the carbohydrate chain, connected to a lipid. You can also have a glycoprotein where we have a carbohydrate chain connected to a protein. Okay. So enzyme, substrate. Here we have the enzyme. This is the active site here. It's not labeled here, but it is the active site. Then we have the substrates that connect to the active site. In the reaction, the substrate is either broken up or it is put together, just depending on the type of reaction that's happening. When the substrate is connected to the enzyme, that's called the enzyme-substrate complex. And then at the end of the reaction, we get the product. Notice that the substrate is changed into the product, but notice that the enzyme remains the same, and enzymes are reused in the reaction, in reactions over and over again. Okay. Here we have a metaphase cell, so we have the chromosomes lined up in the middle. Remember, chromosomes are connected at what's called centromeres. The microtubules or spindle fibers radiate out from the centrioles, and they grab onto and manipulate the chromosomes to get them to go to the right place. Here is a plant cell in the middle of telophase. So we're starting to see the nuclear envelope reforming. The chromosomes are still visible. They're going to start to decondense back into chromatin for just a little bit. There's the cell wall. And then here's the beginnings of the cell plate forming, which starts as vesicles and then turns into the cell plate. Here is DNA replication. Remember, DNA replication happens during S phase of the cell cycle. The replication fork is where the DNA is opened by the enzyme helicase. Then the enzyme DNA polymerase, this here and here, same thing, are going to help add nucleotides onto both exposed strands of DNA. And if you'll remember, DNA is replicated on one side in a continuous direction because the strands face opposite directions. So one side, called the leading strand, is synthesized continuously. So the DNA polymerase attaches on and just continues adding nucleotides. And then in the lagging strand, it's called that because DNA polymerase can only attach on a couple of pieces at a time. So as this adds nucleotides, more DNA is exposed here, and the DNA polymerase has to go back and reform and go back and copy, and that's called the lagging strand, and those pieces are called Okazaki fragments. And our last diagram. So photosynthesis, 2 stages, light-dependent reactions, light-independent reactions. water and radiant energy go into the light-dependent reactions. The water molecule is broken up, and we get the electrons and hydrogens to put on the electron carriers, and we get ATP to move into the light-independent reactions, or the Calvin cycle. So the light-dependent reactions, water is broken apart. When it's broken off, we remove the hydrogens to use those electrons and protons, and then oxygen is released into the atmosphere. Light-independent reactions, or the Calvin cycle, are going to take the carbon dioxide and they're going to do carbon fixation along with the electron carriers and ATP. The cycle is going to spin 6 times, and we end up with a glucose molecule. So the reactants in photosynthesis are sunlight. Technically, it's not a reactant, but in the presence of sunlight, water, and carbon dioxide. Okay? And then the products are the oxygen and the glucose. And if you'll notice, glucose and oxygen both go into cellular respiration. So the products of photosynthesis are the reactants in cellular respiration. So the first stage takes glucose and it breaks it apart during glycolysis, and it splits it into pyruvates. The pyruvates are then going to move into the mitochondria. Oops. First go into the Krebs cycle. During the Krebs cycle, they're broken apart. We release carbon dioxide here. We make NADH, FADH2, we produce some ATP. Then we're going to move into the electron transport chain where we get all those ATPs made. Okay, and from the electron transport chain, remember oxygen waits at the bottom of the electron transport chain and collects those hydrogens and electrons to form the water molecule as a waste product. And that is your review.